My name's Professor Stewart and I work at the University of Birmingham in the UK. And I work in the Department of Cancer and Genomic Sciences. My lab has been interested a long time in inherited diseases in children associated with problems either repairing DNA damage or replicating their DNA in the presence of DNA damage. My up-and-coming talk is about identifying new human diseases and new genes that predispose children to increased levels of chromosome breakage. Then what we try and do is understand the underlying cell biology about how the mutant gene and the mutant proteins actually affect genome stability, potentially predisposing these children to development of cancer. But also because the proteins that are mutated in these children are involved in repairing DNA damage. And it sort of means that the patients themselves are very hypersensitive to the killing effects that you see with chemotherapy. So our diagnostic work and underlying cell mechanisms really helps clinicians to make decisions about what chemotherapy that they can give to these children without getting very toxic side effects. The protein gene I'm going to talk about today was one that we identified from human genetics. It was a gene that had no known function in humans, but from our work and work of several other labs now, it turns out that this gene called donsin is incredibly important, and it actually turns out that it is a critical component of the replication machinery that actually allows cells to replicate their DNA properly. And what we found is that the patients with mutations in these have increased in chromosome breakage because they can't replicate their DNA properly because the donsin protein is mutated. Well, this donsin protein, we only identified it as mutated in human disease in 2017, I think. Very little is known about how it's regulated, how it controls DNA replication, and how it is important in cells for dealing with cells that are trying to replicate their DNA when it's been damaged. Really, we know very little about this protein. Our mechanistic work is really trying to understand how mutations that are inherited in these children actually give rise to disease. But also this gene is mutated sporadically in cancer. So it's of interest to understand what those mutations that have been acquired in these different cancers actually do to actually make the cancer genome unstable. The new area that I was sort of getting interested in is something called suppressor tRNAs. These are a way of modifying specific tRNAs to recognize stop codons in genes to allow the stop codon to be bypassed, and actually you get expression of the mutant gene. This is a potential new therapy in terms of in patients who have inherited disorders, such you could say cystic fibrosis, for example. If the patients have inherited a stop codon, that actually means that the protein produced is lost, or it produces a truncated protein, then actually providing these modified tRNAs, these suppressor tRNAs, they recognize that stop codon. They insert the correct amino acid and allows the normal protein to be produced. So it's a really interesting new therapy for patients that have got devastating diseases, that you can actually bypass their genetic mutation and produce the correct protein.